This is Brain Ponderings with Mark Mattson, conversations with scientists at the forefront of brain research. My guest today is Jeff Macklis. He's professor of stem cell and regenerative biology and also in the Center for Brain Science at Harvard. He also has a number of other appointments. Jeff has both an MD and PhD. He's an expert on how the brain forms, how different types of neurons arise from progenitor cells, and how connections are made, with a lot of focus on the large projection neurons, such as the motor neurons that extend all the way from the brain down to the spinal cord. And he's also made some major advances in trying to develop approaches to replace lost neurons. And of course, this is still a little bit in its infancy and a long way from humans, but it's the idea is in the long run, this basic knowledge will enable that. So Jeff, why don't you just start with, I guess, the pluripotent stem cells, the few cells that form after fertilization of the egg, and then go up to cells that end up in the brain.
Okay, I'm not sure if I'm going to be able to take everybody step by step through that, Mark, but it's nice to see you after all these years. We've known each other and worked in parallel fields, but maybe to pick up on your intro and to take some of that lead. The brain's an amazing organ, as we all know, but as you pointed out from the very beginning of young cellular organization, there appear to be a few cells that start being determined to make nervous system. And as an embryo grows and gets a front and a back, we call that rostral and caudal, those cells start aligning, and the nervous system, through a variety of what we call diffusible morphogens, so chemicals that can spread from one point to others and one point to others and one point to others, they set up this amazing construction organization that anybody who's built a house or leveled a yard will understand. It's finding an exact point within all of these organization centers. And then it starts setting up what many of us in the field, as you know, call progenitors. So there are progenitors for the spinal cord, the more back or rear part of the nervous system, and there are progenitors for the front or more rostral part of the nervous system. And then all of those start separating into different groups. So there are progenitors for the motor neurons in the spinal cord that grow out and connect to muscle. And there are progenitors for the sensory ones that bring in touch from your fingertips. And there are progenitors for all of the lower areas of the brain and for local smaller neurons that we call interneurons. But I'm going to focus, as you said to everybody, on the so-called projection neurons. And at the very forward part of the brain, or in humans, it's the top part of the brain, because we're no longer mice or cats running around on all four limbs, we have progenitors for the cerebral cortex. And I think most of us, even just watching movies, know that that cerebral cortex is that big, loopy, hilly valley structure, and we call those gyri and sulci, the gyri being the bumps and the sulci being the holes in between, the valleys in between them. But we get progenitors that are going to build that whole cerebral cortex. And believe it or not, and Mark, I know you know this, but I'm not sure if everybody else does, the human cerebral cortex is about 18 to 20 inches on a side if we spread it out. It would be like a big fancy cloth dinner napkin. not like a takeout restaurant, but, big one, linen tablecloth and all that. And it's folded up into our skull, pretty much so we can get through the birth canal without somebody dying. And to your point, what that cerebral cortex does is brings in information from the world, processes it, holds our memories and experiences and language and cognition, and turns it into complex motor behavior. And that could either be my hand movements here on the screen or, my language.
Yeah.
And that's what the cerebral cortex does. And the way it does that is that whole 20-inch dinner napkin has literally, in my view, hundreds of little patches, maybe as big as one knuckle, spread around doing different kinds of functions. Listening to vowels, or recognizing a human face, or seeing motion, or seeing color, or identifying if something is new in your office that I haven't seen. And to put all that information together so we can have this conversation, the projection neurons that you were talking about connect. And they might connect from one spot in one side to a similar spot in the other side. They might connect forward to the back of the brain. They might connect, as you were saying, from the motor cortex all the way down through my brain, crossing over down to the spinal cord. So there are these, in my view, thousands, if not tens of thousands of distinct types of projection neurons that set up this circuitry. And I'm almost done this little story to hand it back to you, but this is sort of setting the stage. And that's been the career-long goal that I've had and of my lab, to understand a few things about that whole huge set of very, what we term diverse or diversity of projection neurons, meaning having so many different types that connect and send information, but with real precision. They don't just go out like a spider web and by accident the brain thinks. I mean, you and I, as I'm telling you that story, presumably you're making a picture in your mind from my words. And so are other people on the listening or watching. And that means, and it's true, that all of our human circuitry is remarkably reproducible. It's not exact. It's not exactly the same, you to me or the two of us to somebody else, but it's so close, we can actually think of individual types of neurons. And in the lingo of the biz, we call those subtypes because there's so many types or models of neurons. And my lab's interested in what are the molecules that build all the diversity from here to here, or here to here, or here to here, or here to here, or to the spinal cord. And how do they all grow simultaneously, going through each other and not get lost? So, and I'll get back to that because that has to do with the axons of the cells. And I can mention that maybe as a next part of, as you're asking me other questions. So how do they build? And then the weird thing is, as you know, and as you've spent a lot of your career studying, in neurodegenerative disease, sometimes the whole brain will be normal, except for this kind of neuron degenerates. Why does a disease affect one or two kinds of neurons out of all those thousands? And I think that comes from essentially the assembly instructions. Just like one model of car might run into certain kind of suspension problems and another model by the same car brand doesn't. It's because how are they put together exactly? So we're interested in how they're built or generated, why they degenerate, going the opposite direction. And as you said, if we understand how to build them, how to generate them, maybe we can regenerate them. Thinking like if you know how to build a fancy car, you might know why it goes wrong and diagnose it. You might be able to repair it.
So there's certain progenitor cells that will end up becoming motor neurons, others that will becoming other types of neurons. And you've been working to understand, so the thinking then is that there's certain gene programs, that is patterns of gene expression. Certain genes may be switched on or off and in a progenitor, and then that will make that, when that cell stops dividing and forms a neuron, that will become a motor neuron. So is that the idea? There's like molecular switches.
Yeah, that's a great way of putting it. And just to put a finer point on that, 25 or so years ago, it was thought, maybe even 20 years ago, and in fact, I'll tell you, until a publication of ours just about 14 years ago. So during all that time, it was thought that those progenitors, that to the world, I'll say, some people call those stem cells, but really stem cell means able to give rise to everything. And these are able to give rise to a range of things. So most of us who are being very careful call them progenitors. So the thought was, until 15 years ago or so, that those progenitors divided a final time and made a certain kind of neuron, and then it was done. But now it's clear, and we published the first 2 papers making this clear in 2008, that about half the work is done when the progenitors make a neuron. Then what they've built is sort of a range of kinds of cells. And then about half of the molecular program that you were alluding to is what we term technically post-mitotic. Yeah. So after that division, it turns out there are other molecular programs that take that general kind of neuron through to completion. And if you'll allow me, I'm a very metaphorical person. That's good. And I've always thought that most people in the world don't have experience with the nervous system or with biology necessarily, but most of us have experience, no matter where we live on the globe, with cars. And we know that there are different brands of cars: Chevy, Land Rover, Toyota, Lamborghini, Volkswagen, and also different types of cars. So there might be 2-door sports cars or convertibles or station wagons or 4-door sedans or pickup trucks. And what my metaphor of how this works is the progenitors form, let's say, a Volkswagen, but it's not yet clear whether it's going to be a 2-door sports car, 4-door coupe, or the small station wagon. And they might divide it into a choice of that molecularly versus, let's say, a Volkswagen delivery van or pickup truck. And there are Volkswagen pickup trucks, as you know. And then there's more molecules that then make it precise. It's the 4-door sedan version. And the interesting thing is, other progenitors that are related might make a similar range of Chevys. And so there are divisions at different, to get back to your thought about the molecular programs, these are programs that occur not only in the progenitors, but change dynamically over time, like within the Volkswagens, but also space. So each of those little areas in the cortex might have related progenitors that are making a different range of Chevy neurons, if that makes sense. So it gets very complicated, but there are certain, I think, underlying building rules that build us this immensely complicated cerebral cortex.
And so you mentioned these diffusible molecules that play important roles in the kind of patterning and development early on. And then when you get down to more local environment, there must be environmental signals coming from other neighboring cells or whatever the cell's growing on that influence whether the neuron actually becomes a motor neuron.
Yeah. You know, Mark, I'm going to say with a smile, it's as if we rehearsed this because you're driving it right through. So yeah, exactly right. So after all those diffusible molecules that I mentioned, a few minutes ago, they basically go away. And then we're left with this cerebral cortex that, isn't really a big napkin. It's curved around sort of like double fist. But then along different pathways, you're exactly right. Certain cell types, typically not neurons, typically glial cells of certain types, put out molecules that are like direction finders. And in the business, as you know, we call that, the lingo is axon guidance molecules. And some of them diffuse, and they're almost like a smell, fried chicken smell, come home for dinner, or a skunk smell, go away from me. And there are molecules that draw axons to them or away from them. And maybe I'll get to axons in just a moment. And there are other ones that are not just diffusing, but that are stuck onto tissue. So those are called attached or extracellular matrix bound. So those you might view as like sticky spikes on a fence that say, don't come here, or maybe, a nice lush park that says, come in. And different neurons have different axon types. And maybe I should step back for a minute and explain to everybody what that is. And this, everything so far I've been making up as I go along, but this next one I've thought through this metaphor, so I've calculated it out. For everybody watching, if we're used to thinking about a liver cell or a kidney cell, and if it were the size of my body, which is a little more than 6 feet tall, I would be the cell, the kidney cell or the liver cell, and my nucleus, where the DNA control molecule is, would be like my abdomen, and everything I would do would be on my membrane, and I would make stuff, and it would all be here, right here in my 6 feet of existence. Neurons are wacky. These projection neurons, if their so-called cell body was as big as my 6 foot and a little bit body, they would have something called an axon. And well, first they would have dendrites, which would be like antennas reaching up maybe 5 stories through the ceiling, listening to things and receiving input from other neurons. But the weirdest thing, not 5 stories, they would have an axon, which would be about the size of my arm, 6 inches or so if I'm 6 feet. And this arm would extend, led by something called a growth cone, about 1,000 to 10,000 times my cell body size during development. And in the neurons you were talking about, Mark, that go all the way down to the spinal cord, they would develop about 10,000 lengths, but then they would exist in you and me about 100,000 times longer than my body. So that means they would extend, these axons might extend to the airport downtown, or to a university across town, or across Massachusetts state lines, all the way into Rhode Island, all the way to Providence, Rhode Island, and find Brown University, 70 to 100 kilometers away. And they'd find a target about as big as a typical conference room, like 10 times bigger than my cell body. So that's not a wire. It's actually part of my body. Yeah. So it turns out, if you calculate out, it's almost like, a Marvel comic book, whoever the stretchy man is. This axon is something like 95 or 98% of the volume of my whole body by the time it extends. Yeah. And you're absolutely right. When we start thinking about building those circuits, if I'm that neuron cell body and my axon is growing, as it extends with this so-called growth cone, and it turns out that's what it looks like on a flat dish, But what it really looks like through the tissue as we watch them is more like a little mole. And it's sensing with its little philopodia, its little feet, and it has sensors, and it's smelling almost. It's not really smelling, I should take that back. It's sensing all those skunk odors or fried chicken or spikes or grass, and it's making turns at set places where there are a whole series of maybe hundreds of sequential, almost like a map direction program on, on your smartphone saying, go 2.4 miles and then look for the sign and then turn left. And they literally do that in development. So this guy might go all the way to the airport. This guy of a different type might cross, have exactly the same signals, but say, oh, that's not where I want to go. I want to interpret those signals and go to that university. And that's what my lab has become increasingly interested in recently, as you know, figuring out what are the molecules in those growth cones that make those precise decisions and then set up synapses with other neurons and do that so we can have this conversation.
So there's molecules in the environment as this growth cone moves through that guide them whether they turn or not. There's also ones that can, accelerate or slow down the growth. Then what happens when the axon growth cone reaches a dendrite, say, of a neuron that's going to form a synapse with? Actually, this is something I studied.
Yeah.
When I was a postdoc, I found that glutamate, which is the main neurotransmitter in the brain, more than 90% of the neurons,
yes,
including the neurons you study, use glutamate. And I found glutamate released from an axon growth cone can kind of stabilize the growth of the dendrite and allow further events that lead to synapse formation.
Yeah, that's a great, perfect segue, because everything I've been talking about, and this has been somewhat controversial, it still leads to arguments in meetings about how the fancy term is how deterministic is this circuit building in the brain. And it turns out that it looks like it's quite deterministic up to the point that you mentioned, that the axons building the general framework, going to the right places, and what my lab refers to, and I think many people do, we refer to it because it's a commonly accepted term, the axon targeting is very deterministic. But then, and you know this fellow as well as I do, our sort of grandfather of neuroscience, Santiago Ramon y Cajal, back in the late 19th century, discovered the growth cones and the neurons. And he called these things motile protoplasmic cones of force, the growth cones. And as you were saying, there are also molecules that speed them up or slow them down. And he pointed out brilliantly that growth cones mature into synapses. So how does that work? So as you were saying, if this is the dendrite, like that antenna going 5 stories up of this neuron, and yes, I'm going to give you credit publicly here, all of these projection neurons, as you know, as well as anybody on Earth, are so-called excitatory. They excite the next neuron, and the neurotransmitter they use primarily to do that is glutamate.
Yeah.
So those growth cones are growing. They're finding some synapse of another neuron. When they get there, if I pull it down through the ceiling, they get here and they're to the right place. But then we don't know this yet, but there's evidence in the field to argue for this, that each of these circuit types of growth cones might have a specific kind of address it's looking for on a receiving dendrite. So it gets there, it looks at their various dendrites, says, oh, you're the right type. But then it says, let me try puffing some glutamate on you and try to excite you. And the dendrite isn't just sitting there doing nothing. It's signaling back.
Yeah.
And they act, what the lingo in the field is, as you know, is activity dependent maturation.
Yeah.
So once they start talking to each other, and this just popped into my mind because I used it earlier today when I was exercising, it's like a Bluetooth link. You have this one turned on to be recognized, this one turned on to be a device, but then they have to go, doodle-loop, and they have to link up. Yeah. So it's deterministic about how I get close, but then it's very activity dependent and dependent on what you discovered those many years ago of building the circuitry.
Yeah. Now, let's move on to, so early on you mentioned that in neurological disorders, it's only certain neurons that seem to become dysfunctional and degenerate. So for example, in actually Alzheimer's disease, there's several neurons in different regions, but
yes,
the glutamatergic neurons in the hippocampus are particularly prone. In Huntington's, it's certain neurons that control body movements, same with Parkinson's. And so in thinking about how, and there are, I should tell listeners, there are no treatments that slow the progression of Alzheimer's, Parkinson's, Huntington's. There's nothing. And you'd have to think that in order to restore function, or it could be an injury, spinal cord injury or traumatic brain injury. In order to restore function, you have to somehow get new neurons in there. Not only that, have to get the right type of neuron that will form the right connections with the other neurons that are still alive. And this is something you've worked on a lot. Can you talk about that?
Yeah, and in fact, I'll broaden it a little bit for the listeners to say that there might be, in my mind, at least 4 ways to think about therapeutics for that problem. And just to lay it out clearly, for example, and I can wait, but in ALS, there are certain gene mutations that put people at high risk or high vulnerability. Some of them are so strong that they essentially say you're going to get ALS or Lou Gehrig's disease. Some just raise your risk. But Lou Gehrig's disease, ALS, or in the broader world, motor neuron disease of this type, is specifically 2 kinds of neurons centrally. One that connects from the motor cortex of the cerebral cortex all the way down through the deep brain, down to the other side, down the spinal cord, and connects somewhere down here. And the second is a neuron in the spinal cord, a spinal motor neuron that connects from there out, or here, I guess I'm getting confused watching the picture here, out to my fingertip. So why is this mutation in every cell in the body?
Yeah.
Your eyeball cell, your nose cell, your skin cell, your kidney cell doesn't cause disease, doesn't make you not be able to move your finger when you're born. You might even be Lou Gehrig, the best batter, best to first base, highest batting average, and then develop this degenerative disease in your late 50s typically, or in Lou Gehrig's case, in your late 30s. So what's up with that? It means the nervous system has been built correctly, but then something specific falls apart. And I've thought for a long time that likely has to do with how the specific types of neurons are assembled, meaning they use the same genes, but in different ways. And you, Mark, and I know that there are so-called splice variants. It's sort of modifications or different versions of the same gene, or there are ways that those genes can be modified and have additional parts put on, or it's sort of, again, back to cars. You might have the same starter motor in 20 different Chevys, but it's the Silverado pickup that breaks down because of the starter, because that Silverado needs a heavier duty starter. And the starter's fine. The starter works for 30,000 miles, but it then falls apart. So my lab's been very interested in figuring out what it might be that makes a neuron vulnerable. So before we even think about, I was going to say, before we even think about,
I just point out to the viewers and listeners that turns out many of the gene mutations that cause ALS or mutations in a different gene that cause Huntington's or Parkinson's or Alzheimer's are gain of function. It's called gain of function. That is, the gene, of course, genes encode proteins. So the protein encoded by the mutated gene acquires a property that the normal protein doesn't have. So it's not that the gene product, the protein, isn't working. There's something else. It's doing something it shouldn't be doing.
So Mark, how are you doing with my car metaphors? Is that okay?
Yeah, that's fine.
You raise because you're exactly right. And that's a great thing to point out, because I was talking about starter motors failing as a loss of function. And that's why you wanted to highlight that. Yeah. And I remember reading a newspaper article about 10 years ago about a certain kind of a model of Ferrari that kept on bursting into flames at stoplights. And it turns out they did a recall and they swapped out various things they guessed at, which sort of reminded me of a lot of clinical trials now. They did some guessing, it didn't do anything. They did another recall to the cost of, hundreds or tens of millions of dollars. And then finally they figured out, we don't know why this is happening. And they got the two foremen from the Ferrari factory and they hired them as consultants. And they said, and the foreman said, which exact model is bursting into flames? And they told them the exact model, the 308 AQXR or whatever. And it turns out it was a gain of function mutation that they were using because of the way they wanted to flare the fenders and make it a supercar. They used a certain epoxy that was rated to 700 degrees. And they used it on a body part that got close to the exhaust. And it turns out at about 600 degrees, it heated up enough to vaporize that the exhaust sitting next to it could then burst the fumes into, and so that's sort of a gain of function mutation. It's not the epoxy didn't work, it's that it did something unexpected. And as you point out, a lot of these mutations are leading to aggregates of molecules somewhere. And we're still not sure, people in the fields are still not sure if those aggregates are toxic or if those are trying to save the neuron from something. So, but back to the therapeutic directions, if one thinks that there are specific kinds of neurons that are vulnerable, one approach might be to figure out, that epoxy, let's make sure the temperature runs lower, or let's modify it somehow. So you could get, this information could get you before cellular replacement to preventative medicines, maybe drugs that lower the temperature of the epoxy or that settle down a certain kind of splicing event or a certain kind of metabolic event that puts that kind of neuron at particular risk. So I'm very interested in that because it would be better to be able to save neurons before you have to fix them. A second way is to try to have neurons that are there that have already started to degenerate to reactivate a growth program so that they might work, but maybe in a way that you put in those kind of preventative measures also. So that has the advantage, not quite as good as saving them from the very beginning, but you already have the circuitry built. And then there are at least two different ways of doing what you were talking about and saying nice things about our work over the years. One could think about having progenitors or stem cells in a dish and coming up with that complex molecular program to build exactly the right kind of neuron or close enough and insert it. That has the problem of needing to put it in somewhere, and that's invasive. So my forever dream, and we did some work, as you know, starting 20 years ago, and we're now going to be publishing some new work that, again, I think you properly point out to listeners, is not ready for prime time. It's in mice, it's not ready for humans, but it's about identifying some leftover progenitors of the types that I was mentioning earlier for the different little knuckles on the napkin. And we've been able to identify some of them that are particularly biased to making a class of kinds of neurons. You know, to my prior metaphor, they want to make Chevys instead of Volkswagens. We don't yet have it down to make a, you know, Chevy Camaro, but a Chevy and not a truck. And what we've done is, and what the dream is to take the developmental knowledge and to manipulate those progenitors right there in the brain. So they're in the right place, and they might even have their brother and sister neurons that are still holding on, that haven't degenerated. And neurons do something that you know well called fasciculate. And they find neurons of the same type, and they put their axon around the other axons, almost like a growing vine in the garden. Yeah. So we're interested in all of those: prevention, turning on closely related neurons to grow and rebuild the circuitry, putting neurons in transplantation, although I'm less enthusiastic about that, or recruiting new neurons from progenitors that are there in the brain. But it's a long way off, but we're a lot closer than we were 20 years ago, I can tell you that.
And there are, can you, one of the papers, actually it was more than 10 years ago, and somehow I missed it. So you were able to, in a mouse model of diabetes, you were able to essentially cure the diabetes by doing transplantation and replacing cells in the brain region called the hypothalamus that controls food intake. Could you, I think the viewers might be interested in that study. Could you kind of summarize that?
Yeah, I'll summarize it in just a moment, but before I even do, let me just tell the viewers, this is not a treatment that's either available in humans or that I would advocate for human diabetes, which is really mostly due to cell dysfunction in the pancreas down in my abdomen. This was actually, we used that model to test whether we could rebuild complex circuitry in a complex brain. Yeah. So we teamed up with our colleague Jeffrey Flyer's lab, who works on, the fancy term is energy balance. You know, how to use calories to gain weight, to build fat, to keep lean, to modify energy expenditure. And it turns out that circuitry, as you know extremely well, because you're such an expert in this, is very evolutionarily old. Yeah. It's down in the deep brain. It's not up in the cerebral cortex. And as you just mentioned, it's in the hypothalamus, like hypodermic needle under the thalamus. So the thalamus, and then under it is the hypothalamus. And there is a kind of mouse mutant. And for those viewers who don't know about this, over the last 20, 25 years, the field of biomedicine has learned how to take genes out of mice, put genes into mice, mutate genes, fix genes, do all kinds of things in mice. So we use mice as what we call genetic models. So this was a mouse that was so-called leptin insensitive.
Right.
And without going into all the details for the viewers, it lacked the ability to achieve good energy balance. So the mice became morbidly obese, huge in their body size, even though they were eating the same amount of food. And what we did is went back into development, figured out when the neurons, or probably several types of neurons, it wasn't as precise as I'd like to get, but several types of neurons that control that energy balance. We figured out when they were born during embryonic development. We took them out. So we didn't actually build them from progenitors. We let little embryonic mice build them for us, but then we micro-injected them into the right place. And these ones that we put in were normal. They weren't mutant, so they were normal energy balance neurons. And what happened with the mice is that the neurons grew, they found those targets, they got the right molecular address, they signaled, and they dramatically reduced, didn't absolutely cure, but they dramatically reduced body weight.
I'm going to pause for a second. I got it.
So that was essentially a simple model system in which we could place the correct neurons in. They built functional circuitry and then had this behavioral metabolic therapeutic effect. So
yeah, so it turns out in humans with obesity, typically they have something called leptin resistance. Most listeners will have heard of insulin resistance, and that's when your cells in your body can't respond to insulin and remove the glucose from the blood. So leptin resistance is that the leptin, which is produced by fat cells when you eat, goes into your blood, goes to your brain, tells you you're getting full, stop eating. So the brain of people with obesity doesn't respond to the leptin, yes. And they're hungry, even though, you know, obviously they don't really need that energy. Right. And so this is actually very relevant. Now,
I'm sorry, go ahead.
One thing I was thinking about from a therapeutic standpoint, many viewers will, I expect, heard of CRISPR, even though they may not, some may not know exactly what it is, but there are ways in the case of these gene mutations, gain of function using CRISPR to eliminate the defective gene. And this has been now in many animal models used. And actually there's human studies going on. I think there's at least 2 with success in certain disorders of the blood, but just futuristically thinking, this new technology could, in theory, benefit people with, yeah.
So I'll tell you just because you and I have known each other a long time, and some of the viewers might find this amusing, but we published that, as you know, and you were nice enough to point out, in Science in 2011.
Yeah.
And it was accepted around, I don't know, maybe sometime in the middle or end of the summer, I can't remember. And we thought it was going to come out sometime in October. But the editors at Science decided to hold it for the Friday, the day after Thanksgiving. because they thought that would be particularly amusing to have this overeating phenotype. And it turns out German public radio and the BBC and various other folks thought that was amusing too. So I spent Friday talking with various press corps about this overeating circuitry and energy balance the day after Thanksgiving.
Yeah, that's similar. We did all this work with intermittent fasting over the years and focused on the brain a lot. And I had several things where like they waited till, like we had a New England Journal of Medicine review article and it came out right after Christmas. You know, intermittent fasting. Our people are making New Year's resolutions. So
right. So that's the kind of to get back to where you were leading us, thinking about how do circuits build with precision, can think about how they fall apart in degenerative disease and how you might preserve them or fix them. But it also is highly relevant. And another area my lab has been very interested in, the cerebral cortex, as I mentioned, half an hour ago or whatever, is where our higher level thinking is. So cognition, social interactions, behavior. And it turns out that a number of the genes that we've discovered in mice as being functional in the cerebral cortex are now known to be human risk or causing genes for human autism spectrum disorders and or intellectual disability. Because if that precision of wiring connecting between areas is not exactly right, you'll be a normal, nice, happy, not normal, but a functioning, happy kid or person, but perhaps will have a disability of the intellect, as we call it, intellectual disability disorder, IDD now. And if there are circuits that instead of enjoying this kind of social interaction that we're having, find that to be less enjoyable or even aversive, that might be thought to be underlying autism spectrum disorder, or autism behaviors. And in fact, we're working very actively now on a couple of those genes that we discovered in the brain that were then discovered in human genetic studies to be absolutely causal for human disease. And that's more of a developmental disease. It's not that everything's set up normally and then falls apart. It's that it's not set up exactly right. And I'll emphasize that word exactly. We all probably have friends or relatives of friends who have intellectual disability or autism. And they can be at Thanksgiving and be with us and they're part of the family. It's just, there's something that was less precise about the circuits. So my lab is very interested, not just in the broad targeting, but the precision of targeting and certain extra targeting. You were talking about gain of function before as molecules. We've actually identified that some of this might not just be imprecise circuitry going to this spot rather than that spot, but sometimes another circuit being built, boom, to there that shouldn't ever exist. And that's quite amazing because it's almost like circuit gain of function.
There's a lot of evidence that the problem in autism are as the child is developing in the mother's womb. And there's some evidence that there may actually be just like a general acceleration of the growth in the womb. And therefore, one could imagine that if say the axons are growing faster than normal, that could affect how they wire up.
Right, and that gets to a really subtle distinction that we do in our mice that frequently in those human studies, which we really can't look inside and see what's happening, but it's that a circuit is here today and there tomorrow in the developing brain with the disorder, and normally it would normally be there. And that can either be speeding up, or it can be, as you were pointing out, the lack of the ability to sniff around correctly and slow down.
Yeah.
So in every one of these situations, it's not necessarily just I'm going too fast. It's that maybe I can't sense exactly where I should connect. So I end up too far.
Yeah.
And we're finding that to be the case sometimes as sort of a lack of precision, almost like going down a street in your car looking for the right address on the houses and just having fuzzy glasses. Then you can't tell and you go too far until you see one digit off and then you have to back up. But these things, building a brain is complex. So you got to get it really perfect to yeah.
I'm going to do a
that problem.
podcast on autism, and there is one risk factor that's pretty well established, and that is if the pregnant mom has obesity or type 2 diabetes, that increases the chance that their child probably won't have autism, but it increases the chance they will. So there's something about the environment during development in the womb that seems to play a role.
Right. Yeah, I don't know about those data, Mark, but as you and I know, so many of these associations carry with them many other associations, which might have to do with socioeconomics or other life events or other past historical events. So I'm not sure about all those, but you know those data. And I am going to need to wrap up in the next 5 minutes. So I should hand it back to you to guide us forward.
Well, we covered a lot of ground as we thought about how the neurons cover a lot of ground and and form connections. I think it might be worth just mentioning a couple, or at least one molecule, BDNF. Now, you mentioned that when an axon comes and it's approaching its target, if this neuron is active, maybe it releases glutamate, then that causes the presumptive postsynaptic cell, the dendrite, to send a signal back to the axon. And one of those signals is brain-derived neurotrophic factor, BDNF. Do you have any thoughts on that? And you've studied a little bit BDNF. Another one that affects the growth of neurons is called IGF, insulin-like growth factor.
Yeah, and I think you're raising for the viewers this whole other world that we haven't talked about, right? So I'll use BDNF maybe as a portal through which to view this. And it goes all the way back to Cajal, as you know, in the peripheral nervous system. He and his student Teo, T-E-L-L-O, did beautiful experiments where they took peripheral nerve and saw that if they put it in different places, central neurons would be drawn toward it. So he pointed out, and he coined these terms, that the nerve makes neurotropic, tropic like directional factors, and neurotrophic growth and feeding factors. So you're mentioning these growth and feeding factors, and it's a whole series of molecules, all proteins, peptides, as we call them in the business, as you know, that are derived or made by certain cells, and they provide important, not just nourishment, but signaling that you're in the right place. So in the peripheral nervous system, nerves grow out to muscle or out to various endings, and they use something called nerve growth factor, as you know, the very first of these to be identified by Professor Rita Levi-Montalcini back in the 50s. And that attracts peripheral nerves to their target and nourishes them and consolidates them. And then, as you know, the next one to be identified was BDNF, brain-derived neurotrophic factor. And then there's CNTF and IGF and various GFs, GDNF that I won't get into. But as you rightfully point out, to use my metaphor before, if here's the dendrite and here's the axon, this one might say, I have some molecules, am I talking to the right guy? And this one says, be happy here, and makes BDNF and other molecules. And it sets up almost a mating system of saying, we're going to support this circuit. And BDNF, as you know, has been involved in not only circuit development, but the ability to maintain those circuits, and also more broadly to maintain the health of neurons. And as you were nicely pointing out, almost 20 years ago now, we did some work that individual types of neurons use distinct sets of growth factors through distinct receptors to do different things. So one neuron type might say, oh, I get BDNF, I'm going to grow my axon now. Another neuron type at the same time might say, oh, I'm smelling BDNF, I'm going to stop my axon and grow my dendrite. So it's really important to know how each of these specific diverse neuron types uses those growth factors. And so that's a whole other fascinating but complex area of nervous system development and maintenance.
And I know we got to finish up quickly. Let's make one last comment with these with BDNF. It turns out that physical exercise, like you exercise in the morning, and also doing what you and I are doing now, keeping our neuronal circuits intellectually challenged, those things increase BDNF and actually can increase number of synapses or strengthen individual synapses. So anyway.
Yeah, and I think that's a great point, and you were central in some of that work. And my comment back would be to our viewers, none of us ever think that it's weird to think that if we use our nerves to exercise our muscles, that wouldn't strengthen that circuitry and connections and maintain it. Right. And as you say, Mark, as you and I are having this conversation, we are using all these thousands of different kinds of circuits. And even though we don't feel it, the neurons are feeling it, and they're strengthening that. So I think there's a huge amount of just sort of, it makes sense that your crossword puzzle or your Sudoku or your Wordle or your reading books is going to maintain the circuitry in your brain.
Or hopefully playing chess. I've been playing a lot.
Yeah, well, that too.
Well, Jeff, I really enjoyed talking with you and seeing your face again, and always enjoyed your work, which is right at the cutting edge, and look forward to what's coming up next. So thanks a lot, Jeff.
Great, Mark. It was really nice speaking with you, and thanks for including me in this great project you're doing. Okay, take care. Bye-bye.
Okay, you too. Bye.